Alright, so in a previous video I introduced the class of biological molecules called lipids. So lipids. And I broke lipids down into two main categories. We have hydrolyzable lipids, hydrolyzable, and then not hydrolyzable. And essentially what this means, and we've kind of talked about this, is these lipids can be broken down into smaller sections, and these lipids, these non-hydrolyzable lipids cannot be broken down into smaller sections. And in this, we fit probably the most abundant lipid, triglycerol. And if you remember, triglycerol has the main function in our bodies, at least, of energy storage. So we break down triglycerol because it's a hydrolyzable lipid to get energy. And I want to continue talking about these hydrolyzable lipids, but the other ones that I think are important, instead of having kind of a predominant energy storage function, they have a structural function in our cells and kind of in the biological role. So a structural function. So the first one I want to talk about are phospholipids. So phospholipids. And phospholipids are hydrolyzable lipids that contain a phosphorus atom. So this phosphorus atom usually comes in the form of a phosphodiester bond, and I have no expectation of you to know what a phosphodiester bond is because I don't think we've talked about it. So we start with phosphoric acid, which is H3PO4. That's phosphoric acid, and it has a structure like this. We've got phosphorus double bonded to oxygen and then bonded 3 other times to OH groups. So we have 4 oxygens in total and 3 hydrogens and a phosphorus atom. And that might look a little bit goofy if you're paying real close attention because this phosphorus is bonded 5 times. But remember that phosphorus is a third row element, unlike carbon, so it can bond more than 4 times. It's capable of sharing more than 8 valence electrons. So we've got phosphoric acid right here, and if we take these 2 side OH groups and replace them with OR groups, so let me kind of redraw the base structure here. Still have it double bonded to an oxygen, but if we replace those sides with OR groups, say for example in a dehydration reaction, this becomes a phosphodiester. And it's an ester because this phosphorus is double bonded to an oxygen and an OR group, and it's a diester because it happens twice. So this is a phosphodiester, and this is the form that phosphorus is going to come into play for our lipids. So if we start with triglycerol, which is kind of that basic hydrolyzable lipid, and I guess I better go ahead and draw out triglycerol here. So this is triglycerol, and if we replace this lower fatty acid chain, remember all of these are fatty acids, if we replace that with a phosphodiester, it's going to look like this. So this would be one of the OR groups. This whole rest of the molecule would be the other OR group. And I guess in our bodies, not to confuse you too much, but phosphodiesters at a pH in our body of around 7.4, this hydrogen right here is actually usually deprotonated, so usually carries a negative charge on this oxygen. So we wouldn't have an H right here, and instead we'd have a negative charge. So this right here is a phospholipid. And one of the cool things about phospholipids is, unlike triglycerols, now we have a specifically polar section. So this right here is polar, and then we have a nonpolar section to this molecule. So this is all still nonpolar, and the purple part is polar principally because of this kind of big negative charge and just the polar nature of this phosphodiester, but that allows it to play a pretty cool role in cell membranes. So you've probably heard of a phospholipid bilayer, and what happens is you have a phosphodiester head, so kind of a phosphodiester head here, and then you have two kind of fatty acid tails coming off, and this is polar. This purple part is polar and the orange part would be non-polar, and you get a whole bunch of these and they kind of line up together. The polar heads kind of line up and the non-polar tails kind of line up, and those non-polar tails become attracted to another set of non-polar tails. So non-polar tails and another polar head right here. And this forms a two-layer membrane to our cells. So this is kind of the thing that separates the inside contents of our cell from the outside contents. And this whole middle section right here is not attracted to water because of this kind of nonpolar lipid characteristic. And it keeps water over here and water over here, but separates these two fluid compartments. So that's a pretty neat function, a structural function of the phospholipid. And I don't think you can really talk about phospholipids without stopping at least briefly to mention sphingolipids. They're another hydrolyzable lipid. So sphingolipid. And you need to talk about sphingolipids because they're another lipid that incorporate this phosphodiester unit, but instead of having it kind of on a base of a triglycerol, it's going to be on the base of the amino alcohol sphingosine. So let me kind of draw in sphingosine. And so sphingosine is an amino alcohol. It's got these OH groups, but it's also got a lot of lipid characteristics because it's a really high molecular weight alcohol. This is a lot of carbon here, 15 carbons on this kind of tail. It would extend out like that if I kind of drew them all out. But if we replace this OH group with a phosphodiester, just like we did with our triglycerol, so kind of replace it, it also develops a structural function within cell membranes, but mostly within nerve cells, and I'll show you why. So we've got a nerve cell here with a long axon, and nerves are surrounded by the insulation of myelin. So we've got myelin right here. And the properties of myelin, which allow it to insulate this nerve axon right here, are really predominantly due to the sphingolipid concentration in myelin. So sphingolipids kind of have a similar structural function within cell membranes, but mostly within nerve cells. And then I guess the last hydrolyzable lipid that I really want to talk about are waxes. So waxes. And again, I'll kind of do this briefly, but waxes are also esters, and they're made of a high molecular weight alcohol. So a long carbon chain alcohol that might look like this with an R group, but it'd be really long, so it'd kind of look like this. And then they've got a fatty acid. So a fatty acid, again, really high molecular weight. And if you form an ester out of these two molecules, you end up with a product that looks a little something like this. So a long chain double bonded to an oxygen with an OR group right here. This would be the ester. And so you've got an ester right here, but it's kind of a unique ester in the sense that it's got two really long carbon chains, making it two kind of non-polar sections here. And these two non-polar sections make the molecules very hydrophobic. Waxes are very hydrophobic, and so we see waxes in nature often forming a real barrier against water, and this happens in leaves. We put waxes on our cars to protect the surface of our cars from rain and from kind of the humidity, but waxes are another type of hydrolyzable lipid, and they're hydrolyzable, again, I just kind of want to beat this point home because they've got this ester group right here, carbon double bonded to oxygen with an OR group that can be hydrolyzed. You can do an ester hydrolysis reaction. So that's with waxes. Same thing with sphingolipids. They've got this, sorry, let me make sure you know that that's an oxygen. They've got this ester group right here that you can break down, and you can do the same thing if I go up with phospholipids. Again, we have this this ester bond right here that we can hydrolyze, and we could do it with with these bonds as well, just like we could with triglycerols. So all of these, again, are lipids that can be broken down into smaller units through hydrolysis reactions.